Hello, my name is Piotr Suszyński from Warsaw University of Technology, and I'm happy to introduce you to my work titled A Nextflow Pipeline for GWAS and RVAS with Integrated Test Dataset Generation for Parameter Calibration, which I'll be presenting on a poster session of the ISMB ECCB 2025 conference. My research is centered on the diagnostics of rare genetic disease symptoms. Some of such diseases have very broad range of symptoms, with individuals displaying only a subset of them, and I am focusing on the cases where the reason for this phenotypic variability is unknown. Rare diseases present multiple challenges, and the small sample size is one of them. Genome-wide association studies are a tool which can be used to quantify the relationship between the genetic variants and the phenotypes, but in order to achieve high statistical power, large sample sizes on the order of hundreds of thousands of individuals are required. One of the methods that was developed to be able to find statistically significant associations for smaller sample sizes are gene-based tests, where genetic variants are aggregated on gene or some other level, and one of the most successful tools implementing them is Regini. However, conducting a GWAS requires multiple steps of data filtering, preparation, and transformation, which can be complex and error-prone, but also introduces the question of which filtering thresholds and parameters should be used. For example, we must filter data of too low genotype quality, depth of coverage, missingness, account for possible differences between the characteristics of the data per different phenotype. For example, there may be large regions where depth of coverage is consistently lower for one phenotype than for the others. Try to filter out the sequencing artifacts and errors. We need to account for population structure and ethnicity. Failure to properly conduct the mentioned steps could result in the study yielding false positives, and on the other hand, not reporting true positives because they might got filtered out before the data even got to the association testing stage. Overall, a GWAS consists of dozens of steps, is parameterized by even more parameters, and it's not clear how this should be set. In our work, we're developing a Nextflow pipeline for gene-based association studies, along with a companion tool which is tasked at optimizing the parameters of the associations pipeline. The main pipeline implements all the best practices for the filtering and preparation of data for GWAS, including also data annotation with variant effect predictor and computing principal component analysis to be able to account for population structure. It also includes a model for characterizing the input data overall and per phenotype to be able to work with whole exome sequencing data coming from different study designs, which yields differences in data quality. The tool for pipeline parameters optimization is another Nextflow pipeline which generates synthetic testing datasets of pre-configured characteristics tailored for gene-based tests, runs the association testing pipeline, assesses the results and computes a score metric for each hyperparameter set, which are optimized this way to maximize the ability of the pipeline to recover true positives while minimizing the number of false positives. We hope that our tools will enable successful gene-based association tests for small sample sizes by being able to use better filtering thresholds and pipeline parameters so that we retain more of the valuable data. I hope to receive as much feedback as possible, so if you're watching this before the conference and I managed to spark your interest, please visit me at the poster session. I'd love to have a chat with you. If not, you're welcome to reach me via email.